once in your data set, so obviously the larger your data set, you might have fewer singletons, but you also have more singletons because every individual has some number of rare variants that are unique or at least private or recent mutations. And in extreme, there are these de novo mutations, so mutations that have occurred in sperm or egg cells in the germline, and they're present in a child, but not in their parents, at least not in their parents' somatic blood that we take for DNA analysis. So anyway, we're focusing on DNA sequencing, whole exome sequencing specifically. So what is exome sequencing for those of you that aren't familiar? So conceptually, it's similar to whole genome sequencing, except that we're focusing on the protein coding portion of the genome. That's around 1% of the full genome. It's around 30 to 50 megabases. What are those 30 to 50 megabases? It's basically if you take sort of RefSeq, UCSC, CCDS exon list, you end up with around 200,000 targets, which are actually baited on an exome capture array. And that enriches for exomic sequence, and then you actually just sequence what you've enriched for, those enriched fractions of the DNA. Why are we doing exome sequencing? A, because it's cheaper when you're trying to look at just dollars and cents. It's cheaper when you're looking at 7,000 or thousands of people. For now, that's still feasible. Whole genome sequencing on that scale is not yet feasible, or at least wasn't feasible completely when we had started this. And another, I think, a key reason is often even, you see even whole genome sequencing studies coming out today, people will do whole genome sequencing and then focus their analyses anyway on the protein coding portions of the exome. Not to say there are no important variants outside of the protein coding regions, it's just a lot harder to interpret them. And we have evidence from Mendelian diseases and similar where we think that those should be enriched also in disease associations. Still, even just focusing on the protein coding portion of the genome, the exome, a typical healthy person still has tens of thousands of non-synonymous, meaning non-silent variants that seemingly would impact protein function, and even 100 heterozygous nonsense mutations. And even there's more recent work saying that there's even 20 loss-of-function mutations per, or nonsense or loss-of-function mutations in homozygous form per person. So even though we're focusing on the protein coding portion of the genome, so it's making the search space smaller, the neutral rate or of variation is still pretty high. So even for mutations that we think have an impact, there's a large search space to look for, and it's basically a signal-to-noise problem. And we see that multiple times, both at the sequencing level, but also the biological interpretation. It's something that we have to deal with, just because that's the way the data is. So when you have next-generation sequencing data, including exome sequencing, an important step is actually the processing or sort of data analysis pipeline, but just in terms of you start with the sequencing reads that come off the machine in FASTQ format. There are many different pipelines for calling variation and getting what's known as a VCF file. We're going to focus on really just because that's what has been convenient for us and because of our close collaboration and being at the Broad Institute, using the BWA Picard GATK pipeline. For those of you that are familiar, the basic steps are really mapping the reads to the genome, marking PCR duplicates, calling variants, and then doing some sort of step for filtering variants, or at least marking variants as being higher or lower quality. What I'm going to focus on today is, assuming you've done some sort of pipeline to get variants, how do you actually analyze those variants? So how do we do follow-up quality control to get a more stringently filtered set of data and actually do association with disease in giving two examples and applications that we've done. So the two applications that we actually recently published, one is, both of them are schizophrenia studies. One is looking at over 600 trios, so that's an affected child with schizophrenia, and their two parents, mostly unaffected parents. So that's 600 trios, that's 1,800 individuals in total. And another study was looking at 5,000 Swedish cases and controls combined, around 50-50, 2,500 cases, 2,500 controls. Both of those, again, we're looking at schizophrenia cases when I say cases. And the point of both of these studies was to really assess the role of rare coding variation as related to, as it contributes to schizophrenia, particularly single nucleotide variants and insertion deletions. We're also following up on copy number variation in the exome data, but I'm not going to talk about that today. But really what we wanted to ask, are there particular alleles, genes, or pathways that contribute to risk for schizophrenia, for disease? So I'm going to start first on the Denovo sequencing study, the TRIO study. So conceptually, we start off thinking about what we call putative de novo calls, either a single nucleotide variant, so either like a SNP or an Indel. And what we have is the situation that we're looking for, at least initially, are cases where the mother is homozygous reference, AA, the father is homozygous reference, and suddenly the child is AB, meaning they're heterozygous and there's B allele came seemingly from nowhere, hence the name de novo sort of mutation. So even just looking at that, we're going to focus just on the left plot. It's pretty similar to the right plot. The left plot here is just looking at SNPs. This is looking at Indels. So on the x-axis, we have here the total number of heterozygous SNP calls that an individual has. We actually sequence the samples in 3 different batches at 3 different sites with different exome captures. That's why they cluster differently. But what you actually see is overall there's a pretty good correlation here between the total number of SNP calls and the number of putative de novo calls, these sorts of scenarios where the individual has a SNP that isn't present in either of the parents. And there's a bunch of outliers. Even after removing those outliers, though, if you look at the scale on the y-axis, you see that the average here might be something like 100. In fact, it's like 164. So if we just would take the raw SNP calls at face value, SNP and Indel calls at face value, each trio would have around 164 de novos, which is way too high. And I say it's way too high based on a bunch of previous studies looking at whole genome sequencing and particularly whole exome sequencing, where the average you expect is really around 1 per trio. And other studies have dealt with this as well, but I'm just going to detail the protocol we used for filtering these 164 on average to get down to the 1 on average. And what we actually did was used our own data set to QC these de novos. And what we did was, if this was around a few 100,000, these putative de novos, which are really just exquisitely enriched for false positives, what we wanted to use was our own data and sequencing metrics to compare to what we call the transmitted singletons. So what those are, we have 600 trios, that's 1,200 parents, 2,400 chromosomes, everyone's diploid. So in those 2,400 chromosomes, if there's a variant that's observed in exactly one of those parents, in one parent, and transmitted to a child, that's sort of an intrinsic validation. So we wanted to use these transmitted singletons really observed twice in our data, but only once in the founders and the parents, and use these 180,000 variants as the positive controls of what we expect higher quality variants to look like, and compare those to what these putative de novo calls look like. So what we're going to do is, in this case, you have a TG SNP, so there are G alleles on these, G bases on these alternate allele reads, seemingly, and the question is, do these reads have certain properties in de novo mutation, or what look like de novo mutations, to be able to filter out instances of false positives? So basically what we've done, and these are just the key metrics that we've looked at, but we've looked at others, and they'll be listed on the next slide in our paper, is just even looking at depth. So if you look at the number of sequencing reads that map to that particular site, we see that the green, which is the distribution of transmitted singletons, really has much higher depth than these putative de novos. It's really just, if you just take the calls, the call set is meant to be very sensitive, which works if you want to be very sensitive, but you're also very sensitive to noise, and this is really just enriching for noise when you're looking for these cases where the kid has an allele that the parents don't. So if you look at the putative de novos, they're just very low depth. Similarly, if we actually look at these genotype likelihoods, which are basically a statistical quantitative measure of assessing how likely it is to have the particular genotype, in this case the heterozygous genotype, we see that these de novos tend to have really low likelihoods in the model, and that model takes into account the base qualities and other things, other metrics, sequencing metrics that are part of what comes off the data, what's in the data that comes off the machine in the FASTQ files. And we see that the transmitted singletons have much higher likelihood. So we're going to want to filter on these metrics. Another important metric is, in a perfect scenario, you'd expect around half the reads to have the reference allele, half the reads to have an alternate allele for a heterozygous SNP. And we see that's basically the case, or at least there's a distribution around 50-50 for the green, for the transmitted singletons. For the de novos, again, there's both very few numbers of reads and a very low fraction of them being alternate reads. So we see that they're really just systematic biases in the data that we need to account for when we want to call de novo variation. So what we're going to do is filter on these and similar types of metrics, but these are really the key ones. So again, if we start off, we're looking at a bunch of different metrics here. It's not important right now, but the point is, even if we just look at the main metric of the number of de novos per trio, and this is focusing only on the single nucleotide variants, if we have, without any filtering, if we just take the raw calls that come out of the GATK sort of pipeline, we have 100 over 100 de novos or seemingly de novo mutations per trio. Once we start adding the depth filter, the genotype likelihood filter, the allele balance filter, which is looking at that ratio that should be around 50-50 in both the parent and the child, or respectively in the child and the parent, it should be homozygous, so it should be close to 1. And some additional filters, we get down to the number of around 1. So these are sort of metrics that we expected, and that's really what we focused on. Can we get the number of de novos per trio down to a more reasonable number using these metrics that really indicate systematic biases? And there's other sorts of supports here that we were doing the right thing. Then we went ahead and validated those de novos. We have around 1 per trio. We have 600 trios. We have around 600 de novo mutations. So one of the things that people have noted many times before is when you're looking at mutations, transitions, that is purine to purine mutation or pyrimidine to pyrimidine mutation, are a lot more common than transversion mutations. And that's due to the biochemistry and particular methylation, particularly at CG sites. So what we actually see is that if we're looking here at all possible, what we've done is mapped out all possible trinucleotide contexts, base contexts, and single base pair mutations. So these are all 192. With reverse complementarity, it's around 90, it's 96. So just looking and sorted by their frequency in our data set, so we see that 36% of de novo single nucleotide variants are actually CG transitions, which is basically what we'd expect. But if we break it down here, what these bars are actually saying, the non-red parts are really just saying these are recurrent. So these are de novo mutations, these particular bases, and those exact genomic locations are actually observed in databases, either in another study that I'm going to talk about soon, the controls, or just in dbSNP in the database. So really these recurrent mutations tend to happen mostly at the CG sites. The 86% of these non-red de novo mutations that we found are actually at these CG sites. So these CG sites tend to be or a big chunk of the mutations, and most of the recurrent ones are in this chunk also. And so this is going to be a little important later. I'll mention why. But also, just in total, around 12% of de novo mutations that we found and validated in our study as being true de novo mutations actually appear in dbSNP. So if you want to ascertain the full sort of set of de novo mutations in your sample, you can't just filter on dbSNP and say, well, that's not a real variant, because we actually saw that even after Sanger validation, the rate is actually pretty high. Going back to association with disease, what we next asked was, are the number of mutations increased? So we know that the average is around 1 per person, but still there might be some outliers. The answer was there really weren't any outliers. There are no particular individuals that had more than 5 de novo mutations, and overall the distribution looked basically as expected for a Poisson distribution with that sort of average of a little over 1. So in terms of association with disease, we didn't see any huge signal or some subset of biological outliers even that might indicate that de novo mutations contribute to schizophrenia. Still, we can then ask, are there particular types of variants? So we could just take the codon table, right? That's why we're doing exome sequencing, so we could focus on protein coding variation. We can ask, are there variants that are silent, missense, nonsense, frame shift, splice site affecting? Can we look, are there particular classes of variants that are enriched in schizophrenia cases as de novo mutations? So we could group them into non-synonymous variants, which are just looking basically excluding silent mutations from the coding set. Or we could look at the more strictly defined loss of function set or disruptive set, which is really just focusing on things that are guaranteed to, or seemingly guaranteed to have an effect on protein function, including nonsense, frame shifting indels, or essential splice site mutations. Again, these aren't guaranteed, but they should be enriching for things that are more likely to have an impact and perhaps be more associated with disease. When we did that actually here, what we see is that we look at controls and we look at the rate of loss of function mutations. Here we're just normalizing it by the loss of function to missense ratio. But in any case here, if you just focus on the bars, we see that in the schizophrenia studies, there might have been an increase compared to the controls, but this wasn't significant. And this was in contrast to what we saw in the literature from the autism and intellectual disability de novo mutation studies. And even though intellectual disability had many fewer trios, they found a very marked increase in the number of loss of function mutations, and the same is true in autism also. So basically what this is telling us is that the rate of de novo mutations and the rate of loss of function mutations in schizophrenia samples in particular wasn't increased. But still, we can still ask, while the rate might not be increased, are there particular genes or gene sets that tend to have de novo mutations in schizophrenia more than you'd expect by chance? So one of these gene sets that we focused on was that we went into the study with sort of a prior hypothesis were these postsynaptic density genes, which are highly sensitive to neuronal activity and signal transmission. I'm not going to go into detail about them now. Just the reason, just to give you some background, the reason why we were interested in them is we actually saw in a previous study by our group that Denovo copy number variation, where you delete, or where there's deletion or duplication of larger portions of the genome, were actually enriched in schizophrenia and tended to hit these postsynaptic density genes. This is from a paper in 2012. What we actually wanted to do was model, do the Denovo single nucleotide variants hit these postsynaptic density genes more than we'd expect by chance. So if you sort of think of the genome, and to do this, we built this tool called DNenrich, or Denovo enrichment. So if you think of the genome, you have all genes here, and then you have some genes having sequence coverage, right? We don't capture perfectly all genes, so we focus on the genes that have sequence coverage. But then we also know that longer genes are going to be more important in terms of de novo mutations, only just because they'll at least have more de novo mutations, it's because there are more bases to be hit by de novo mutations. And we need to take that into account. But as we saw earlier also, that there are certain base contexts, like in this case, let's say, just symbolized here by the CG positions, certain genes might have more CG in them, and you'll want to take that into account because those genes will have more mutations, and genes with fewer CG positions will have fewer mutations just because of the sort of base composition of those genes. So we want to take that into account when we model the genome. And the reason we're modeling the genome and not just using controls is we don't have a large enough set, no one has really a large enough set to fully know what the probability of having a mutation is, or what the background probability of having a mutation in a particular gene in the genome is, because we don't have enough control trios yet. We'd need tens of thousands, if not more, really, to estimate the per base mutation rate at every single base in the genome. So what we do is we think of the de novo mutations as sort of darts, and we're going to throw them at this dartboard, this genomic dartboard, but we're going to throw the darts in a way that recapitulates where they fell originally, but in different genes. So if it hit a CG site in one particular gene, it will have to hit another CG site, but it will be scattered among the genes and their CG sites at random. And we're going to do that 10,000 times. We're going to do permutation, basically. So we throw the darts at the genome, and then we can ask, do the darts hit particular genes or gene sets in the observed data? Yep. So does it hit the genes in the particular gene set more or less than we observed in practice? And we actually can say that there's enrichment in candidate genes, including these postsynaptic density genes, by looking at this, using this framework. Another example of what we do is we also look at overlap with autism and intellectual disability, and we also see that there's significant overlap. And I'm going to skip those examples. Moving on very quickly to the population, the second study, we'll go through this quickly. One of the important things we wanted to look at was, are the case and controls well matched with respect to technical attributes of the data set? And this is important just so that you know that any case control analysis you do downstream is well matched. So we looked at depth, number of variants, things like that, and none of them were significantly different between case and controls overall, which is good. That's a technical sort of QC. We know that we knew from previous studies that the case and controls were well matched for common variants, despite having some Finnish ancestry even in the Swedish sample. But we also wanted to check, are they well matched with respect to these rare genetic variants? So even just as an example, if you have a doubleton, so you have a variant observed exactly 2 times, it can either be shared both by a case, by 2 cases, shared by 2 controls, or in one case and 1 control. And we really saw that the number of variants that are shared by cases, by controls, or in a case and a control is really well matched to what you'd expect by chance, and the same was true when you start looking at other sets of rare variants. So moving on, we actually want to just do association in these hundreds of thousand variants in 5,000 people, and you could split it out the way we did before, but even just focusing on these 100,000 variants, what's important to know about sequencing data is because you're looking at rare variants, what you actually have is, if you think of it as a 2x2 table, most of the time you just have a 1 to 0, meaning the variant is observed, it's a singleton, it's observed exactly in 1 case or exactly in 1 control. So you're never going to have statistical power to analyze that type of information, so what you need to do is group the variants together, and people do that using gene-based tests, and there's lots of different gene-based tests that take into account different things. I'm not going to go into detail about that. I did running out of time. Even when we look at gene-based tests, 1 gene at a time, we actually don't find any significant gene, and no significant single gene. So even in 5,000 samples, we don't find a significant gene. But what we next thought was, can we take, if we've grouped the variants into genes, can we now group the variants in the genes into gene sets and ask for enrichment? So what we did was we grouped around 2,500 genes. We sort of collated that list from prior studies that were whole genome studies looking at genome-wide association, copy number variation, and including the Denovo study that I mentioned previously. We ended up with around 2,500 genes. That's around 10% of the 20,000 genes. And we asked, is there overall enrichment in that set? And the answer was yes, there is overall enrichment. Here it's broken down by the contributing data sets, and we get a pretty significant p-value, 10 to the minus fourth, for this type of data set. Asking, though, where does that burden actually lie, we see that it's really driven by these singleton, these rarest variants when they're aggregated, looking at nonsense variants that are seen exactly once in the population. We combine them across these 2,500 genes, and we start seeing burden. What we don't see is any burden in the missense variants. It doesn't mean it's not there, but in terms of the power that we currently have, we really don't see any signal in the missense variants, and that's even when we've stratified by different predictions of what these missense variants and their impact might be. We don't see any sort of burden in cases versus controls in silent variants, or even more common but still rare variants. So really it's telling us that we need to focus on the singleton nonsense variants to see a burden. Last slide. So what we actually see is that the burden also represents, we looked at 2,500 genes, but really we see that the burden represents rare mutation across many genes. So it's really not localized to a few genes. This is what you'd expect, this sort of plot, if it was localized to 25 genes, let's say, a very small number of genes. What we see is that it goes up really slowly, the burden, as we add the genes. So really, it's really localized, it's really diffuse across the multiple genes. Conclusions, we saw that schizophrenia de novos can point us, de novo mutations can point, if you carefully filter them and test them, it can point us to particular processes. And in the rare variation in the population, we saw that the rarest, most deleterious variants, the singleton nonsense variants, in a broad set of genes, are associated with schizophrenia, but we still can't point to any particular gene, probably because of the effect size, and we still need larger samples, I guess. That's really our conclusion from that. Obviously, there are lots of people involved in the studies. I'm not going to list all of them now, but I will list our ongoing software that we're developing sort of in an ongoing fashion is this PlinkSeq software that basically we've developed and used to run most of the analyses. Thank you.
The usual process, please step up to the microphone if you want to ask a question.
Hi.
Hi.
So I just wonder, for the case control study, presumably there's cryptic de novo mutations in there.
Yep.
So how much of the signal that you see for the nonsense would be explained by the population incidence of cryptic de novo mutations?
So it's possible that those singleton nonsense mutations are enriched for de novo mutations. Right now, it's a little bit harder to tell. One of the things we're actually doing is using identity by descent to try to narrow down where we think there might be more recent singleton mutations, that they're not just private, that we can sort of date them, how recently they occurred, and that would basically be a generalization of de novo mutations, even when we don't have the parents. That's ongoing work, so I can't tell you what fraction of those might be de novo or at least recent yet, because we don't yet have the answer. But you're right, that's definitely something to follow up on. Thank you.
Thank you. It's a great talk. I want to ask a question. How many mutations, de novo mutations, you would expect in trial?
Entirely?
No, for each individual.
Each individual?
Yes.
Well, in the exome, what people have found, and that's what we found as well once we did the filtering, you find around one per
Only
individual. One exome mutation. So it's really, you're starting with, yeah.
If you are doing whole genome sequencing, how many would you expect?
So there was a paper by Decode 2 years ago where they looked at that, and there's now a paper by Genomes of Netherlands project. The answer is around 70 to 100. So if you're looking at 1% of the genome, you're seeing around 1. If you're looking at the full genome, you see around 100 times, not exactly, but something similar.